the way we handled it, we recorded a 25 minutes presentation run and performed by different specialists from the Knauer team. And after that, we will have a lot of time for questions and we'll move on to this topic. And maybe first I ask you kindly, if you haven't done so already, please mute your phones or your microphones and your cameras, as you probably experienced in the last couple of weeks as well. Internet performance might vary, especially when a lot of people are using their cameras and their microphones. And Knauer started already, I think, almost 2 years ago to apply at German legal authorities to get approval to work in our application lab with cannabis. It was a long way we had to go before we got the official approval. We even had to establish a safe in our lab where we keep the samples. And so this is all under strict control. So we made a lot of experience on this topic. And we started first with our application team on analytical methods, and Lisa Loxterkamp will share her experience on and using analytical HPLC for the quality control of cannabis. Knauer offers analytical and preparative HPLC solutions, and there are different options available if you also want to use purification methods, chromatographic purification methods for the production of cannabis. This can be done in a batch process with preparative HPLC or even with a continuous process. We made here experience also together with customers and learned here a lot. And we just put our major, let's say, experiences or overview together in a 25 minutes presentation, so that you get to know the different possibilities to use chromatographic solutions in handling or working with medicinal cannabis. My name is Anke Börtgen, and I'm head of product management and marketing at Knauer, and I'm very happy to accompany you as a moderator through this webinar. As mentioned, Lisa Loxterkamp from our application lab will start with describing analytical HPLC methods in quality control of cannabis. After that, my colleague Max van Delbrück, who is product manager for the preparative LC, he will describe our Cannabis Purifier, how this can be easily used for batch processes. And after that, Paul Peach, who's a product manager from Continuous Preparative HPLC, will describe if you want to go even for scale-up processes. After the presentation, we will handle questions via the Teams chat. If you have already a question during the session or the presentation, please write them in the chat. We will try to go through it from top to bottom. If time runs out, we allowed, of course, more time than the 45 minutes. Somebody has to switch off microphone, please. Okay, so all microphones, please be switched off. And so we will then handle the questions via chat. After the webinar, you will receive via email the recorded presentation as a link. And if you have questions afterwards, please just send them to us. Okay, I think we are ready to start, and I ask our technical expert to start the presentation right now.
Hello and welcome. My name is Lisa Loxterkamp. I'm an application scientist at the company Knauer, and I would like to tell you something about our Cannabis Profiler today. But first of all, if we want to talk about cannabis, we need to have some terms in mind. At the top, we see the medical cannabis, which is produced in regulated environment. This means we have certain growing conditions. Variation within the medical cannabis are different contents of THC, for example. In some countries, you only have access to medical cannabis when it's prescribed. Next is the hemp. In comparison to medical cannabis, hemp is used in the agriculture or industrial sector. and only contains a low amount of THC. In Germany, for example, it's only allowed to grow hemp with a content below 0.2% THC. Last but not least is the recreational cannabis, which is the illegal form distributed on the streets. Wrongly used are the terms marijuana and hashish in this case. Marijuana and hashish are not per se illegal, they are just a processed form of the original flower itself. Marijuana is the dried flower and hashish is the pressed resin. But what makes cannabis so special and interesting, and why is everybody talking about this? Well, the interesting part lays inside the flower, the ingredients, not only terpenoids, but cannabinoids playing the important role here, displayed here. There are more than 100 known cannabinoids, for example, shown here only 16 of them. These cannabinoids are found in large amounts in most plants and are therefore of great importance. The most important cannabinoid is probably the delta-9 THC, the delta-9 tetrahydrocannabinol, which is not only responsible for the drug effect, but also regulated under the narcotics law. In Germany, you need to have a permission to work with this kind of cannabinoid. Looking deeper into the structure, you can see always an acid form and a neutral form from each cannabinoid. This results from the plant itself. The plant produces only acid forms, which get converted to the neutral form by heat, for example, smoking or drying the flower. The crucial part with cannabinoids is that the interaction with all of them is important for the effect. If you want to know more about that, look after the entourage effect. I talk now about drug effects and narcotics law, but what are the effects of cannabinoids? As an example, a few years ago, approximately 5 years, I watched a video about a patient with Parkinson's disease. As you know, there is no cure for this disease, just things to make life bearable. This person in the video got so sick he couldn't even talk properly. Then he tried smoking cannabis for the first time, and the effects were tremendous. No body shaking and normal talking. So cannabis can be used as a drug in a lot of diseases, but still there are not enough clinical trials to confirm all medical application areas. But still, cannabis is a huge opportunity. That's why a lot of countries make it legal to prescribe. Even in Germany, we now grow our own medical cannabis and expect the first harvest at the end of 2020. What does this mean for us? Well, quality control and monitoring are the essential keys, and that's where the Cannabis Profiler comes into play. And here it is, the Cannabis Profiler, consisting of 4 different modules. From the top, we have a low-pressure gradient pump able to support 4 different solvents, followed by the multi-wavelength detector able to detect 8 different wavelengths. And for high throughput, the use of an autosampler is crucial. The last module is the column thermostat for constant temperature. So what does the Cannabis Profiler offer you? For the cannabinoid content, different matrices can be analyzed, such as flower, seeds, or oils. Another advantage is that we offer the method for determining aflatoxin in cannabis flowers by changing the configuration of the system. An additional fluorescence detector and a derivatization unit makes it possible to analyze aflatoxins. Quality control regulated in a pharmaceutical environment needs to follow pharmacopoeia rules. For this case, we offer the DAB method with a qualification and quantification of 6 different cannabinoids as shown on the right side. For the more research area, we additionally offer a method to determine 16 different cannabinoids. The quantification is conducted by 228 nanometers, and the differentiation of the acid towards the neutral form of the cannabinoids can be seen from the 306 nanometer absorption. So, to summarize, with the Cannabis Profiler, you can act flexible for the use purpose and HPLC to conduct different analytical issues.
Welcome to this webinar also from my side. My name is Max von Delbrück and I'm a product manager for preparative systems at Knauer. I would like to draw your attention from cannabis analysis to preparative applications with cannabis. In short, this section is about obtaining vessels with pure cannabinoid at the end of the HPLC run. You already know this chromatogram. Lisa presented it in the earlier analytics section. It shows the separation of 16 cannabinoids. We have developed a purification method for a prominent cannabinoid, namely cannabidiol, or in short, CBD. An established process for the extraction of CBD from hemp plants is oil extraction. Since cannabinoids are fat-soluble, they dissolve in oil and are thus extracted from the plant material. Although hemp plants with high CBD content provide a high purity of the cannabinoid in the extract, some applications require an additional purification step to remove impurities. Preparative HPLC is the method of choice here. This slide shows the chromatogram of a preparative HPLC run with CBD oil as a sample. The two marked fractions have been analyzed with the already mentioned Cannabis Profiler. Let us first take a closer look at fraction 1. On the left side, two chromatograms of the analysis are shown. Blue-colored is the signal of a run with CBD oil before purification. Here, several peaks are shown, which indicate a mixture of cannabinoids. Red highlighted is the chromatogram of a sample of fraction 1 after purification. Here, a dominant peak is visible, peak 2, which corresponds to CBD. A much smaller peak is peak 1, which corresponds to CBG, which co-eludes with CBD. Remaining cannabinoids and impurities present in the blue chromatogram were successfully removed. The analysis suggests that preparative HPLC can be employed to produce CBD of high purity. Similarly, fraction 2 of the preparative chromatogram was examined. The red signal trace comes from a sample of fraction 2 where only one peak is visible. Peak 3 corresponds to CBC. The data show that the developed method is very well suited to separate CBD and CBC. This example purification illustrates that individual cannabinoids can be purified by preparative HPLC. According to our application specialists, there's nothing to prevent a method development for each individual cannabinoid. In the following slides, I would like to introduce the Cannabis Purifier, the system which was used to develop the just mentioned method. The ternary gradient pump is equipped with a valve block which allows to form a gradient from up to 3 eluent containers. The sample is injected via an injection valve with sample loop. The cannabinoids are detected after the column at 228 nanometers by a single UV detector. Finally, a valve for fractionation is placed, which allows to collect the separated cannabinoids in 11 fractions. If you find that 100 milliliter per minute are no longer sufficient, you can easily and cost-effectively upscale the system while continuing to use the devices. The better parts, such as the pump head, flow cell, and valves, are exchangeable components and can be replaced with larger variants. For example, the 100 milliliter pump head can be replaced with a 250 milliliter pump head, or valves for 1/16 inch capillaries can be replaced with variants for bigger capillaries. If flexibility at module level is important to you, the Cannabis Purifier gives you all the freedom you need. In this configuration, the upper system component is equipped with one UV detector and 2 valve drives, one for the injection and the other for the fractionation valve. The combination of modules can be customized. You can freely equip this HPLC docking station with detectors, valve drives, and pumps. In this combination shown here, a UV detector is installed on the left, a valve drive in the middle, and a pump on the right position. The HPLC docking station offers maximum functionality with minimum space requirements. The Cannabis Purifier is controlled by a touch-optimized software mobile control, which is installed on a tablet. The tablet is attached directly to the system. Since no PC with screen and keyboard is necessary, the space requirement is minimal. The software is intuitive to use, so the training period is short. Basic purification tasks can be solved with the function for threshold-based fraction collection. Depending on the value of the detector signal, the target substances are automatically collected in different vessels.
Hello everybody and welcome. My name is Paul Pietsch and I'm the product manager for simulated moving bed chromatography. In this video, I want to introduce our cannabis producer, the Azure SMB. Simulated moving bed chromatography as a continuous LC method sets the standard for productivity, purity and yield in continuous chromatographic processes. And this strength can be also used for the purification of cannabinoids. First, I would like to explain short the SMB process before I go into detail about our cannabis producer. The simulated moving bed chromatography is explained by the theoretic concept of true moving bed process. In contrast to the classical chromatographic processes in which only the liquid phase moves, the solid phase moves in countercurrent. The principle of the TMB is illustrated in this figure. You see a standing column where the liquid and the solid phase moves in countercurrent direction. The process room is divided by the feed and the eluent, the two inlet streams, and the two outlets, extract and raffinate, into 4 zones. The mobile phase flows from zone 1 towards zone 4, here from the bottom to the top, and the solid phase from zone 4 to zone 1. Feed, in the example here a binary mixture of the components A and B, is continuously added between zone 2 and 3. The more adsorbed component A moves with the solid phase here downwards and can be removed as extract between zone 1 and 2. In contrast, the weaker adsorbing component B is transported with the liquid phase towards zone 4 and can be removed as raffinate between zone 3 and 4. In zone 2 and 3, the separation takes place, and zone 1 and 4 have the function to regenerate the solid and mobile phase. A technical implementation of the TMB process is hardly possible. These circumstances led to the development of the SMB process. The SMB is divided as the TMB by the two inlet streams, feed and eluent, and the two outlets, raffinate and extract, into 4 zones. The solid phase consists now of columns connected in series. Usually per zone 1 to 4 columns are used. Technically applied is the periodically switching of the in and outlet in the flow direction of the liquid phase, and this causes the simulated movement of the solid phase. The process concept is limited to the separation of binary or pseudo-binary mixtures, since separation is only possible in the two fractions extract and raffinate. Typical areas of applications are the separation of chiral components in the pharmaceutical industry and the separation of glucose and fructose in the food industry. And the important question now is, of course, can I use SMB for my cannabinoid purification task? And I cannot give you a generally applicable answer to this, because we must have a look on the separation task and especially on the chromatogram. Okay, then let's have a look now on an example chromatogram. A classical SMB separation is a separation in 2 fractions. Here you see a typical chromatogram which literally screams for an SMB application. You see a binary mixture, baseline separated, and this mixture can be separated in 2 fractions, raffinate and extract. The big strength of SMB is that binary mixtures do not necessarily have to be baseline separated. The peak fronts can overlap, which is usually the case, for instance, for chiral substances. And nevertheless, separation with up to 100% purity and 100% yield can be reached. Of course, mixtures of natural products are mostly not binary mixtures. such as CBD oils. If this is a chromatogram of a CBD oil, and the aim is to purify this single component here, which elutes in the middle of other different substances, then in this case, the best process is a preparative single column chromatography with our Cannabis Purifier. However, if you want to purify a particular cannabinoid or a cannabinoid fraction, then it is of course possible to separate a multi-component mixture with SMB. For example, a late eluting cannabinoid, like in this example here, could be purified in the extract fraction, and so it can be separated from the other early eluting components. Or the opposite, an early eluting cannabinoid or cannabinoid fraction can be separated from the late eluting substances. Then let us summarize what are the decisive prerequisites and also advantages from SMB to batch. For example, if you have your cannabinoid separation task ahead of you and SMB is an option. First, you have a binary or most possible pseudo-binary separation task. And second, your separation has to be isocratic. But especially the unbeatable argument for SMB application is that separations at once with high purity and high yield are possible, and only about 10% of the eluent consumption compared to a batch chromatography is needed. This sounds interesting, or? Then let's have a closer look at our SMB systems. We as Knauer have technically implemented the SMB process as follows. Here you see again the flowchart of the SMB process. The Knauer Azure SMB consists of 4 pumps and 7 multi-position valves. The devices are arranged as follows. 3 pumps, extract, raffinate, and eluent are placed inside the SMB cycle, the feed pump outside. 4 valves are placed at the pump outlets and 3 valves at the pump inlets. Due to this configuration, the SMB system can be used very flexible. For example, the number of columns can be adapted without any hardware change, just with a click in the software. We have an 8-column setup with 2 columns per zone. Here on the right side, you see our lab system, and we recognize our assistant ASM 2.2L. The system consists of 4 ASM, each equipped with 1 pump, here on the right side, and multi-position valves. The system is very compact and finds its place on every lab bench. The max pressure of the system is 130 bar with a continuous flow of 30 milliliter per minute. We offer the system in 2 versions, one in stainless steel and another one in a biocompatible version. Then we come to our cannabis producer system. And here I would like to cite SMB specialist Christian Ulbricht. Currently, the SMB is the only purification process that can be duplicated on a large scale for the purification of cannabinoids. You see a photo of our cannabis producer here on the left. This SMB system has principally the same design as the lab system. The system consists of 4 preparative pumps P21L and 7 multi-position valves integrated here into 4 Azure Assistant ASM 2.2L. The 7 multi-position valves are operated by the valve unifier VU 4.1, and the valve unifier provides the newest technology to our SMB systems. The smart valve drive features automatic valve recognition and adjusts torque and switching speed specifically for each valve. Especially useful for a continuous mode, the FID enables to read GLP data. For example, the maintenance of the rotor seal exchange is simplified by automatic notification. This is where the advantage also of the ASM 2.2L comes into play, which Mr. van Delbruck has just presented. Especially in continuous production process, you can have 1 module on stock, and in case of service, the user can quickly exchange the modules by loosening just 4 screws, and therefore the downtime is reduced to a minimum. While the system continues to run, you can take care of the maintenance of the affected module. The pilot SMB system includes 4 flow meters, allowing a complete real-time monitoring of the entire SMB process, and the system can also be equipped with detectors. The system is available up to flow rates of 400 milliliter per minute with a max pressure of 200 bar. As a complete system, as you see here in the picture, the cannabis producer will be pre-installed on a table. Every device and capillaries are fixed and therefore well structured. The product streams and the column in and outlets are placed user-friendly outside the table. The product streams here on the right side and the column in and outlets on the left side, ensuring a fast and easy connection via tube fittings. The easy cleanable stainless steel surface, anesthetic wheels, and the leakage tubes fulfills main requirements for production areas. If you know of the opinion, the system seems quite complex, and how do I actually control this? And that's where our user-friendly software, Purity Chrome MCC, comes in, developed based on our preparative software. And here on the left side, you see the typical SMB flowchart, and the pumps and the 7 multi-position valves can be controlled by this visualization. You're also relieved to calculate with our integrated SMB operation point calculator. The SMB parameters will be automatically calculated on the parameters of your preparative run. And if you're interested in the system, but you do not have any previous knowledge of SMB, or you doubt that you can operate the device, or you need support from us, we offer different SMB services. For example, method transfer and optimization, or an SMB expert can help you at the first days to start with your system. If you want to get more familiar with the SMB theory, I invite you to visit our Knauer Academy in Berlin and to take part in the SMB Beginners course. I hope you got interested in the Knauer SMB system, and I'm looking forward to hear from you.
Thank you, Lisa, Paul, and Max for this presentation. And I think maybe there are some questions. I have a couple already, and maybe if it's okay, I just start and go ahead. I know at Knauer we spend a lot of time on sample preparation. Lisa, could you please share your experience? Which grinding preparation do you recommend before the extraction?
Yeah, that's no problem for me. So it's a little bit depending what kind of throughput you have. So if you have a big amount of samples, then I would recommend that you're using devices like ball mills with different kind of vessels. So you have like up to 50 different vessels for the sample preparation or the grinding. But if you're only doing development, it's sufficient if you're using mortar and pestles. But you need to think about that the cannabinoids are kind of hydrophobic. So if you're using hydrophobic containers or vessels, the cannabinoids can stick onto the surface. So I would recommend that you're doing the grinding in the same container as the extraction. So you have the whole cannabinoids out of the container. So that's what I would recommend.
Mm-hmm. Very good. And does anybody in the audience has a further question on this topic? If yes, please just write in the chat. Okay, do you have further questions? I'm asking the audience now again regarding the analytical methods Lisa shortly presented. Good, maybe then thank you, Lisa. Please also later on, there is more time for questions. Thanks so much. And maybe also something related to our purification system. When applying a batch purification with the Purifier Max, how can larger sample volumes be applied?
So in the shown example, purification 2 milliliter were injected via the sample loop. Of course, there are different sample loops. The largest one is 40 milliliter. But if you have really large samples, you can use own sample pumps. So when you think of the assistant module, the HPLC docking station, you can easily exchange modules. So you would exchange the valve drive with injection valve and insert a sample pump, which can run up to 50 milliliter per minute. We have also examined the version to inject the sample via one channel of the main pump. So this is also possible.
Thank you. And that means I can start with lower volumes for sample injection and later on upgrade the system.
Sure. The Purifier is highly modular, so you're free to exchange the modules or even the valves to bigger valves. So you can still use the basic devices. Yes.
Mm-hmm. Very good. And thank you, Max.
You're welcome.
Does anybody from the audience has additional questions to the purifier? If so, please send a question in the chat. Okay. Then maybe a further question. When we move from batch purification to a larger scale with a continuous process, it's a big step. You have seen the systems displayed by my colleagues, and the SMB system, the pilot system, is of course a very big system. And Paul, could you please recommend what are the basic steps to develop an SMB method, like an SMB method, or rather, how do I actually start?
Yes, of course. Generally, I start with an analytical separation to meet the prerequisites for an SMB process. As you heard further, this is you have a binary mixture or pseudo-binary mixture. The method has to be isocratic. Also nice is to do it with room temperature, because later you need to scale up and to avoid heating your columns. And in this analytical method, you, for example, screen different columns, different illuents to meet these prerequisites. So if you have your analytical separation, or a lot of customers already have the separation ready, you scale up to a preparative or semi-preparative scale. You overload the column, and that you still have a separation, and the selectivity of both analytes or both fractions between the both separating peaks has to be around 1.2 up to 2.0. And with this preparative column, you can optimize the method a bit more. And important is also that the separation material in this preparative column is the same you will use later for the SMB. And another task is there that you finally determine the absorption isotherm of your analytes. And this is really easy in the linear range, that you determine the retention times of the two peaks, separating peaks, and the dead time of your column. And with these parameters, you go into our software and you can calculate your SMB parameters. This would be the basic steps.
Mm-hmm. We got a question for you, Paul, and I read it out loud. So regarding the remark, avoiding heating the column, could you please explain what you were hinting at?
Yes, because I mean, you want to go in the pilot scale in a little, I would say, production range. So avoiding heating the column, if it's possible, it's good, because heating means energy means extra cost. So if you can do a separation with room temperature, I would prefer it, because later if you want to scale up, extra heating would mean extra cost. Also, what we can recommend to do a separation with SMB and normal phase, because later if you want to, if you have your product, you want to concentrate it, so you want to evaporate a bit of a solvent, and this is, for example, easier to evaporate a normal phase than, for example, a reverse phase. You would need less energy and that means West Coast.
Sophia, did that answer your question? I'm looking at the chat. I hope so. If not, yes. Okay. Are there any further questions? I'm just checking the chat. Okay. Yeah, thank you very much for my colleagues for the presentation, and you will receive the recorded webinar via email. And if you have questions later on, please get in touch with us. So Knauer has a direct sales team in Germany. Worldwide, we are working with partners. We have very long established partnerships. If you're not sure who is your contact in other countries, please check our website. There's also, I thought there's a further question. No, but I think that's it. So if you don't know who you should get in touch with, please check our website or respond to the email of the webinar. Thank you very much for listening. And in the next couple of weeks, we will present more webinars. Due to the time of Corona pandemic, many of us are in the lockdown. I can say for Knauer, we kept our operations running, our production and our deliveries, and we are happy that you are here with us and that we can move on to develop new methods and technology for medical reasons or a better life. Thank you so much. Bye-bye. Ist abgeschaltet?